Hi, my name is Alisia Tumac, and today I will present on tumorigenesis, immunotherapy, and the role of adjuvants. As you all know, cancer remains a major cause of death worldwide, its annual toll being around 9.5 million deaths in 2018 only, with 17 million new cancer cases annually, according to the International Agency for Research on Cancer. Carcinomas can be broadly divided into two groups: metastatic, which are the principal cause of cancer-related deaths, and non-metastatic. During metastases, disseminating cancer cells escape from primary tumors and acquire cellular traits that basically allow them to travel and colonize distant organs and form metastatic tumors. Both primary and metastatic tumors are complex ecosystems composed of neoplastic or abnormally dividing tumor cells, extracellular matrix, and accessory non-neoplastic cells, which can include support cells, endothelial cells, and also infiltrated inflammatory immune cells. I mentioned inflammatory for a reason. During tumor formation, the tissue evolves into this highly specialized microenvironment that contains corrupted extracellular matrix and essentially presents chronic inflammation. Cancer is believed to be caused by unresolved inflammation, in fact, and the impact from inflammatory mediators can both initiate and eliminate tumor cells and also prevent tumor development. So what does this mean? Well, there are two types of inflammation: chronic inflammation, which is associated with the development of cancer. One example of this is long-term tobacco consumption or smoking. This can lead to chronic inflammation in mouth and lungs, respectively. And studies show that you are more likely to develop oral or lung cancer when you're a tobacco user or a smoker. On the other hand, acute inflammation is associated with tumor regression. And one example of this is vaccines. Vaccines are able to basically recruit and activate inflammatory immune cells short term to create long-term memory cells. Next, I want to talk about the different types of tumors. Tumors can be homogeneous or heterogeneous. Homogeneous tumors under selective pressure from cancer immunotherapy are completely eradicated. On the other hand, heterogeneous tumors form from immune-resistant cells that survive treatment and end up repopulating the tumor with heterogeneous tumors. This type of tumor is much more difficult to treat than the homogeneous type tumor, and I will discuss how exactly we get to have a heterogeneous tumor in the next slide. During the early stages of tumor development, both the innate and the adaptive immune cell response recognize and eliminate the more immunogenic cancer cells, and this is called the elimination phase. In this sense, the elimination phase is the ideal immune surveillance action, where initially transformed cells can be cleared by the body's defense system upon tumor antigen detection. However, knowing about the heterogenic nature of most tumors, we can see the equilibrium phase and the escape phase are actually more common. In the equilibrium phase, the immune system gradually loses domination and basically allows neoplastic cells to survive in a dormant state. These neoplastic cells continue to evolve until they reach a clinically detectable tumor, as seen in the escape phase. And different subsets of inflammatory cells can, in fact, impact tumor fate. Selected cancer cells that survive progress to a clinically detectable tumor can adopt strategies of what is called peripheral immune tolerance, and they end up recruiting immunosuppressive immune cells that can subdue other tumoricidal cells. Due to this phenomenon of peripheral immune tolerance, and also due to the immunosuppressive nature of tumor cells, cancer treatment can prove extremely difficult. The first step towards coming up with the right immunotherapy treatment is understanding the cancer cell immune cell crosstalk in metastatic tumors and immune surveillance. Let's say these guys are part of a cell line. You have one happy guy, a weird guy. You can't tell if this guy is dangerous or not. And the mean guy, you know for sure this guy is dangerous because he starts shedding tumor-associated antigens, and he also doesn't present as an MHC or major histocompatibility complex 1. And I will explain this. Antigen-presenting cells and even natural killer cells have the ability to sense abnormality in proliferation and also sense released tumor-associated antigens. The tumor cells shed TAAs, which essentially get engulfed by and displayed on the surface of the antigen-presenting cell as a MHC1 or 2. Typically, tumor cells will fail to display MHC1, and because of this, a strong immune response can be induced involving cytotoxic lymphocytes. Antigen-presenting cells will prime T helper CD4 cells, which in turn will release cytokines to activate CD8 cells, natural killer cells, and macrophages. The cytotoxic CD8 T cells are the most prominent anti-tumor cells. Upon priming and activation, these cells differentiate into cytotoxic lymphocytes, or CTLs, and CTLs and natural killer cells are both able to exocytose perforin and granzyme-containing granules, resulting into an acute inflammation. Perforin is a substance that basically pokes holes in the cell's membrane, and granzyme is used to induce cell apoptosis. And finally, tumor necrosis factor alpha basically recruits more immune cells over at the site of inflammation. The cool thing about CTLs is that following exposure to a target cell, they're able to undergo massive clonal expansion, and then they're also able to contract to about 5% remaining cells, and this 5% will become memory T cells. This is an ideal situation because memory T cells can further recognize and differentiate back into CTLs if they encounter more cancer cells of the same kind they just killed. We now know that the T helper 1 immune response in tumors correlates with a favorable prognosis in terms of overall survival and also in terms of disease-free survival in many malignancies. The T helper 2 immune response, on the other hand, although favorable for pathogens, is not favorable here because B cells activation and proliferation have been associated with chronic inflammation, and we now know that the chronic inflammation correlates with tumor growth. But if T cells are so effective at killing malignant transformant cells, how do some cells manage to overcome the attack by these effector T cells? Typically, in a T cell dependent process, most neoplastic or abnormally dividing cells will express highly immunogenic antigens, such as this guy, and that's what gets them recognized and killed during the early stages of tumor development. However, guys like this one who secrete some or no tumor-associated antigens will adopt an immune-resistant phenotype. So let's say this guy survives the APCs and the natural killer cells without getting recognized, and now he's in the equilibrium phase, but once he begins to divide, this cancerous cell achieves the escape phase. During tumor development, cancer cells evolve mechanisms that mimic the phenomenon I discussed, the peripheral tolerance, and are able to prevent the local cytotoxic response of effector T cells, natural killer cells, and others. Peripheral tolerance also means that T cells response is regulated via immune checkpoint to protect tissue from inflammatory damage, and tumor cells really want to be protected from T cells because cytotoxic T cells are essentially their major enemy. So they have the immune checkpoint blockade with CTLA4 and PD1 acting as negative regulators, and they also use T regulatory cells to downregulate T cell activation. However, remember, cancer cells thrive when chronic inflammation is present, so they can also corrupt tumor-associated macrophages and tumor-associated neutrophils to help boost an immunotolerant microenvironment by secretion of immunosuppressive molecules such as IL-10 and tumor growth factor beta. And essentially, macrophages and neutrophils will contribute to the chronic inflammation and help tumor growth. They also inhibit I want to mention, they also inhibit the secretion of interleukin-12 by dendritic cells, which we've seen in the previous slide. It helps with the T helper 1 response. So they inhibit the T helper 1 response. So everyone's immune system uses the same method to attack cancer, but the process is more complicated than the process of recognizing and killing a regular pathogen, because cancer cells are created by your body. Because of this, the normal ways to find and fight invading cells from outside the body aren't always effective. If the body can't tell the difference between tumor cells and normal cells, then the tumor cells may be able to hide from the immune system. So I like to think of the allergy shot analogy. Whenever you are given allergy shots to relieve the symptoms of an airborne allergen, such as pollen or pet hair, increasing the doses of a specific allergen injected into a person over a series of visits will cause the body to essentially develop a tolerance to pollen. This type of therapy can provide temporary or permanent relief of symptoms. However, the body no longer sees pollen as an invader, so the immune system stops attacking it. The case with cancer cells is often similar. In early stages, cancer cells may shed proteins into the body. As these proteins circulate consistently through the bloodstream, the body begins to develop a tolerance for cancer cells. And once that tolerance exists, the immune system may not recognize these cancer cells as a threat. Then, just like pollen, the cancer cells may be safe from immune system attack. So these immune system mechanisms are important to mention, because in order to come up with an efficient cancer treatment, you have to understand the crosstalk of different tumor-associated immune cells in the different stages of cancer progression. Immunotherapy is a type of cancer treatment that, as its name suggests, it helps improve the immune system's response against cancer. And over the last several years, there have been great advances in the field of cancer immunotherapy, in the field of cancer immunotherapy, due in large part to a greater understanding in T cell signaling and regulation. In particular, the use of T cell checkpoint inhibitors such as anti-CTLA4, anti-PD1, or anti-PDL1 revolutionized the care of patients for melanoma, lung cancer, renal cell cancer, bladder cancer, and so on. And amongst others, it proved effective against radio and chemotherapy resistant tumors, which is great. However, or checkpoint inhibitors can have adverse reactions, because even though you want active T cells, too powerful T cells can benefit the tumor treatment, but they can prove cytotoxic and detrimental to the patient long term. The ability, the ability to utilize surface modified, inorganic, organic, or even combined nanoparticles shows many possibilities to apply nanoparticles in drug delivery for cancer treatment or image contrast agents, and also for diagnostic purposes. The plus part is that they are biocompatible, able to modify gene and protein expression, and are also able to track immune cell infiltrations. They can also, however, they can also display rapid clearance, poor targeting, and toxicity in certain areas. And these are their limitations. However, due to known and displayed immunogenicity of nanoparticles, and especially in the case of virus-like particles, they can often initiate an immune response on their own, which can be beneficial for the treatment outcome. The third and the category I will be focusing on today in detail is the use of immunoadjuvants for cancer immunotherapy. An adjuvant comes from the word aiuvare in Latin, which means to help or to enhance, and is basically defined as any compound used to enhance immune responses against antigens. It is commonly used for regular vaccines, and now it has been also introduced into cancer vaccines and treatment. In 2010, the first anti-tumor vaccine, Sipuleucel-T, was approved for the treatment of advanced prostate cancer. This vaccine was adjuvant adjuvanted with what we call GM-CSF, which is granulocyte macrophage colony stimulating factor. It's a very long name, but basically is a cytokine that promotes lymphocyte production. In 2015, an oncolytic herpes virus was delivered as an in situ immunotherapy and was approved for the treatment of melanoma. In situ vaccinations mean, or in situ vaccination represents an alternative approach in which a cancer cell can be, a cancer vaccine is generated in vivo without the need to previously identify and isolate a tumor-associated antigen. Basically, instead of engineering cells in a lab, scientists can use radiation or injection of a special virus that kills some of the cancer cells and stimulates the tumor to release their tumor-associated antigens, which were previously hidden inside the cells. You're essentially creating a personalized vaccine within the body. In 2017, the first immunotherapeutic gene therapy was used using autologous T cells, which are cells obtained from the same individual as the one that's receiving the treatment. Further, CXCRs are basically transmembrane G protein coupled receptors, primarily responsible for growth and movement of cells during angiogenesis and neovascularization, which is the fancy term for development of new blood vessels. And this happens whenever a tumor forms. So the development of CXCR antagonist limits migration and metastasis of an existing tumor and also helps improve immunotherapy outcomes. So you can say this is an adjuvant form of therapy rather than talking about an adjuvant itself. The same thing is with focused ultrasound heating, photon radiation, and laser treatment. There are also forms of adjuvant therapy. They basically cause cell stress and antigen release and therefore help promote cancer recognition by immune cells. The limitation with these adjuvants is limited evidence of clinical translation. I mean, this is expected since coming up with an adjuvanted regular vaccine is already hard enough, coming up with the cancer vaccine after understanding the immunology behind it is even harder. And so it often depends on the mice models used. Different criteria may be used to group adjuvants. Adjuvants can be classified according to their physicochemical properties, origin, and mechanism of action. You can see there are lipid adjuvants, nucleic acid, mineral salts. You have proteins, microparticles, and you can even have combinations of 2 or more adjuvants. And what they do is these adjuvants will act on toll-like receptors or nod-like receptors to induce some sort of immune response. The difference here from regular adjuvants, and it's highlighted in red, is the use of cytokines for cancer vaccines, such as we've seen in Sipulus LT vaccine earlier. And we can also have modified viruses, which fit in the nucleic acid section here. And the difference here is that regular adjuvants are basically synthetically made single-stranded RNA, double-stranded RNA, or single-stranded DNA. But in this case, you're introducing a live virus or a live modified virus to help with cancer treatment. So antigen-presenting cells and macrophages are a big part of the innate immune response, and they can pick up and display antigens on their surface. Antigen-presenting cells also have pattern recognition receptors, more specifically toll-like receptors. They help recognize foreign antigens. So how does this work with adjuvants? Adjuvants will exploit the PAMP and DAMP associated molecular patterns. And in the case of PAMP, you can have an adjuvant that mimics a pathogenic bacteria or virus and will bind toll-like receptors on immune cells, and it will stimulate the immune response, which will be accompanied by inflammation, hopefully acute inflammation, activation of adaptive immunity, and eventually will proceed to resolve the infection and allow for tissue repair. The plasma membrane receptors, the plasma membrane toll-like receptors, recognize extracellular looking pathogens, while the internal or endosomal lysosomal receptors can recognize intracellular or intracellular-like pathogens. So adjuvants mimic bacterial or viral infections by introducing specific components that will activate the immune system through these pattern recognition receptors. Another type of pattern recognition receptor is a nod-like receptor, and this is related to the DAMP associated molecular pattern, or basically tumor cells will barely release any tumor-associated antigens, but instead may secrete a damage-associated molecular pattern when they're dying. So this can be either from an external or an internal factor. Typically, it's due to laser radiation therapy or chemotherapy, which will cause cell death, and it will cause the cancer cells to release these damage-associated molecular patterns, which will bind to the antigen-presenting cells and will cause an immune response. So immune cells that participate in these processes include antigen-presenting cells, macrophages, as well as T cells and neutrophils. Both damage-associated and pathogen-associated molecular patterns may also stimulate adaptive immunity and participate in autoimmune responses and tissue repair. So one can use a combination of DAMP and PAMP adjuvants, or can only use one adjuvant versus the other. It's very common to use both because you want to cause cell damage to cancer cells by using a damage-associated molecular pattern, so that you can initiate the immune response and cause some APCs to migrate over. And then you want to have a PAMP adjuvant present at the site of inflammation, such that it will amplify the immune response further once these antigen-presenting cells will recognize it as such. Next, I want to talk about the different types of adjuvanted cancer cell vaccines. You'll find that these are a bit different than the regular vaccines. So you have three types: preventive, therapeutic, and neoantigen vaccines. The preventive vaccines are basically similar to regular vaccines. So they will immunize against the viral infection that is responsible for development of cancer cells. For instance, cervical cancer and head and neck cancer can be caused by human papillomavirus, or HPV, where liver cancer can be caused by hepatitis B virus, or HBV. This type of cancer vaccine is very similar to a regular vaccine, again, because it's used to preventively immunize against the pathogen that may cause cancer. Next is therapeutic vaccines. This vaccine came from the fact that each individual tumor is in some sense unique and has its own distinguished antigens. This is also why tumors in different parts of the body or organs may require different treatments. As a result, more sophisticated cancer vaccines approaches are necessary. And fortunately, doctors can now identify targets on patients' tumors that help distinguish cancer cells from normal cells. Sometimes these targets are normal proteins that are produced at an abnormally high level by cancer cells. And one example is prostatic acid phosphatase, or PAP, which is often overexpressed by prostate cancer cells. So researchers have taken advantage of this insight, and they came up with the Sipuleucel T vaccine, which they adjuvanted with the cytokine, but used the overexpressed protein as an antigen. Additionally, virus-derived proteins expressed by virus-infected cancer cells offer another promising source of markers that can be targeted through vaccines. Another example is a a tuberculosis vaccine that acts as a general immune stimulant. It actually became the first immunotherapy of any type to be approved by the FDA. And how it works is basically BCG is put into the bladder through a catheter, and it reaches the cancer cell and turns on the immune system. In order for this to work, the BCG actually has to come in contact with the cancer cell. And so the immune system cells are attracted to the bladder, and then hopefully once they get there, they will recognize the cancer cells. Again, in this category, we see one vaccine that is antigen adjuvant combination and another that relies on the bacteria itself as an adjuvant to act on the pathogen-associated molecular patterns and hopefully elicit an immune response that will recognize the bladder cancer cells. And third category and last are neoantigen vaccines. In order to create an individualized cancer vaccine, researchers had to identify cancer-specific peptides called neoantigens, and then use a cell, protein, or nucleic acid-based platform to deliver those neoantigens to patients in order to prime or activate the immune systems to attack the tumor. Antigen-presenting cells, such as dendritic cells, will internalize the cancer-specific peptides and then look for and select for the major histocompatibility complex proteins. So these triggers, and this is shown better in this slide, this triggers T cells with receptors that bind those neoantigens to basically differentiate into effector T cells that will mobilize an immune reaction and hopefully kill the cancer cells. So I'm going to briefly explain the neoantigen vaccines. The first type is the dendritic cell vaccine. What they do is basically extract monocytes from the blood of patients, and they culture it with synthetic versions of selected cancer neoantigen, and they form a mature dendritic cell carrying those neoantigens. These cells are then reinfused into the patient's circulation. And in this case, you can say that researchers are adjuvanting the immune system by introducing already primed dendritic cells that will cause an activation in innate and adaptive immune response. Next, you have long peptide vaccines, and these are just synthetic peptides containing the neoantigen sequences that are injected into the body, where they are picked up by antigen-presenting cells. And then third, you can have DNA and RNA vaccines, and then how this works is nucleic acids encoding for neoantigens are introduced into the body, where they are translated into proteins and picked up by antigen-presenting cells. So this vaccine in itself can act as an adjuvant by also acting on endosomal toll-like receptors. Next, I will talk about oncolytic viruses and vaccines. So recent oncolytic virus development resulted in discovery of novel viruses, which can be engineered to, they're actually not novel viruses. So they're viruses that are modified or engineered to induce powerful tumor-specific immune response. In addition to their use as cancer vaccines, oncolytic viruses have the advantage of being directly cytolytic to cancer cells and promote antigen recognition within a highly immune-stimulating tumor microenvironment. So this is a special type of cancer vaccine in which you basically introduce a live modified viral strand that is able to replicate in tumors, in tumor cells only. Although oncolytic vaccines' efficacy was initially attributed to directed killing of tumors, scientists have now discovered, and it's kind of obvious that the oncolytic viruses themselves will also induce an anti-tumor immunity because they are viruses and they are immunogenic. So you can say that the oncolytic virus itself is acting as an adjuvant. Last, I want to talk about cytokine adjuvants, and in particular about one.
Hey, Alisia.
Yes.
Can you go back to that previous slide? My question is, are the immune cells or T cells, are they recognizing the cancer cell as the antigen, or is it recognizing that the cancer cell is infected, and so that's why it's causing it to lyse?
That's why it's causing it to lyse.
No, like yeah, like is it because once it's infected, they're recognizing that the cell is infected, and so the T cells are attacking it because it's infected, rather than recognizing the cancer cell's antigen change.
So kind of both, because the virus itself is causing because it's dividing inside, like it's causing the cell to lyse, and once the cell lyses, it releases all the tumor-associated antigens that are inside. And so since you have a surge of those tumor-associated antigens, you're attracting immune cells over. And so once immune cells come over, they will also see the virus, and so the immune response will be amplified. Does that answer your question?
I guess I wonder more because I know that in some cases, after the immune system clears the infection, the tumor cells begin to grow again. And so the immune cells were able to, I feel like the immune cells are not recognizing the cancer cell as the infection. They're just getting crossed more in the crossfire. And so I feel like, I don't know if it's actually, causing the T cells to respond against the antigens from cells, but I'm not entirely sure about that.
So that is possible because cancer cells can mask themselves as self cells. And so the immune system may recognize like the primary tumor, but then again, like if the tumor is heterogeneous, like I explained earlier, then you may kill part of it, and then some cells may be able to hide from the immune system and then grow again into like a second tumor.
Hmm.
Hey,
Okay.
I have one thing. So as you know, immune system, the cancer cell or tumor avoid the recognition by the immune cell. So I don't think that the immune system can recognize cancer cell. That's the problem, because I think if you could be able to recognize that one very good, so you could remove that, but it's not very, possible to recognize the cancer cell or tumor by itself. This is what I read based on what I know.
Yeah, that's true.
So I want to complement this slide. That might be completely wrong, and kind of talk about what Michael said, because we know that, for example, like cancer cells often have impaired anti-viral defenses, right? So that's why this particular platforms are good. Like viruses can penetrate, promote cell lysis, and in the process, all the antigens will be released, right? That's when you create your immune response. That's why these are called adjuvants. But essentially, those antigens, once these are released, the immune system will recognize and create a memory to those antigens. And eventually, that memory can be used for attacking other newly formed or not detected cancer cells. Am I correct, Alisia? Because I think that's where this is so potent.
That's the hope with these adjuvants.
Okay, because I know this is has become very an area of wide research of investigation because of that. And apparently there's some great results. But anyways, sorry for the interruption.
I had but wouldn't the local you said earlier that the local inflammation could cause an immune. So if all of these tumor cells are released in a fast, what's the word I'm looking for? Like really quickly, we see this inflammation occur really quickly. So then wouldn't that also cause, like maybe the immune system doesn't see the cell itself, it sees the inflammation, maybe?
I mean, inflammation is due to immune cells coming over and then secreting more cytokines to call more immune cells over. And so that's how you start inflammation. So yes, this would be a type of acute inflammation. So I said earlier that cancer cells secrete a limited amount of tumor-associated antigens. And so the immune system may be blind to those tumor antigens, but whenever you have like so many tumor-associated antigens from the lysis of these cancer cells, then you have immune cells started to come over to the tumor. And so hopefully they will recognize the other tumor cells that weren't lysed. I mean, that's the hope with this treatment. And then hopefully they will be even more activated because of the presence of a virus in the tumor cell environment.
Yeah, that makes sense.
That makes sense? Okay. Any more questions on this slide? Okay, I'll go on to the last type of adjuvants that I want to talk about, and that's cytokine adjuvants. And basically combining vaccines with cytokines seem logical, but it can also bring unwanted toxicity. And this has been observed with the use of interleukin-2. In addition, the non-specific activation and expansion of unwanted cell subsets like regulatory T cells, which can be immunosuppressive, can contribute to a global immunosuppression and limit vaccination response. And despite this, a variety of preclinical models demonstrated the potential therapeutic benefit of combining interleukin-2 with vaccines, leading to the study of interleukin-2 in clinical trials. The anti-tumor effect of interleukin-2 is actually believed to be mediated by lymphocyte expansion and augmentation of T cytotoxic cell function. The FDA approved IL-2 as a single agent for use in patients with metastatic renal carcinoma and also metastatic melanoma. And interleukin-2 has also shown efficacy as an adjuvant for infectious vaccine therapy. So keep in mind that studies vary and depend on the type of vaccine administered, the IL-2 dose, the timing of IL-2 administration in relation to the vaccine, and also the length of therapy. And although initial clinical trials using interleukin-2 as a vaccine adjuvant demonstrated some modest effects in the immune response, new combination immunotherapies use gamma cytokines to promote natural killer cell and T cell expansion, and also effector T cell function. And these are promising strategies to enhance the immunotherapy of tumors. A number of different vaccine strategies have shown potentiation with interleukin-7, interleukin-15, and interleukin-21. And you can see here the effects they have on the immune cells. Moreover, interleukin-12, which is not shown here, but which I showed earlier that is used to start the T helper 1 immune response, is also on clinical trials currently. So in conclusion, I talked about tumorigenesis, I talked about immunotherapy, what it is, what it does, and different kinds. And I want to conclude by saying that adjuvants are preferred in cancer immunotherapy due to their cost effectiveness, effective innate immune signaling, good immunomodulatory capacity, high specific antibody production, and antigen-specific clonal expansion, along with cytotoxic cell formation, contributing to long-lasting adaptive immune response. And it's also helpful in lowering the cost of antigen production by making the antigen more potent while using less of the antigen dose. Thank you. Any questions?
That was great. No, that was actually very, very good. Certainly a level for which everyone can aspire to in their own presentation. So basically, you have on your summary an interest in having a good humoral response, which seemed to contradict what you said early. Or I'm sorry, you said good antibody response, right?
Oh, that's my bad.
Okay, so that's yeah. So what
That's
you're
my mistake.
if I had to summarize, essentially what you want in a good cancer immunotherapy is basically a Th1 response and
Yes.
almost no Th2 response, right?
Yeah, you don't want Th2 because you can induce chronic inflammation.
I see. Okay. Do you know of any checkpoint inhibitors that are small molecules, or are they all protein?
I thought they were all proteins, but I'm not very familiar with checkpoint inhibitors.
No, I assume they mostly are. And just these adjuvants that they're using, they're mostly of, it sounded like human origin. Like, what was that rather long one you were talking about? Macrophage something or other. Those are human macrophages?
It was like a granulocyte macrophage colony stimulating factor.
Yes. Is that a human factor?
I am not sure actually.
Hmm. Okay. Okay. Well,
They're using a lot of like cytokines and primed immune cells, like primed dendritic cells.
okay.
Adjuvants.
And so the thing that ultimately ends up killing the cancer cell are those natural killer cells or CD8 plus T cells?
You want both, but more of the CD8 T cells because CD8 T cells are specific. Natural killer cells are non-specific, so they just kill like basically if you don't have a major histocompatibility complex 1 or if they feel like you're dividing too fast. But
Right. So you said at the beginning that cancer cells don't express MHC1, right?
yes.
So why aren't natural killer cells just killing them?
I mean, there's also, I think it's, I'm not sure. I mean,
Yeah.
they can, but they're not that visible to the immune system in a way.
Yeah. Okay. Well, yep. Anyway, nevertheless, I really learned a lot. So if anybody else has any questions, go ahead. And
Hey, Alicia.
Hey, Michael.
From all you're reading, if I wanted to get into this field, what do you think is the most promising immunoadjuvant?
What do I think is the most promising?
Yeah.
I think cytokine therapy is pretty cool, but it can also be dangerous.
How come?
Well, because you risk of you have the risk of inducing non-specific immune response, and if you do that, essentially if you mess up, you can get chronic inflammation, and then you can use immunosuppressive mechanisms instead of the response that you want.
Hmm.
I don't have favorites.
Hey, Alicia, I have a question.
Yes.
Okay. So at the beginning of your presentation, you mentioned that some of these tumors, if I remember correctly, are constructed from a convolution of heterogeneous cells. Am I correct?
Yes.
Okay. So let's say, based on your reading, let's say that you're using an adjuvant, for example, the oncolytic adjuvant, and whenever you have this heterogeneous tumor, chances are that you have like growth factors. Let's say, for example, in the breast cancer, you might have like collagen and stuff that will only allow the virus to penetrate into a certain extent. So based on your reading, do you know of how this oncolytic treatments are coupled with some other, for example, therapeutics?
Are you asking me if you can couple an oncolytic virus with a different therapeutic?
Yeah, like for example, let's say that they use oncolytic viruses to kind of decrease the population of cancer cells, and then they complement this with something else. If that's possible, and maybe like what do they complement it with? You know?
I know that you can partner it with, I believe, T-cell therapy,
Okay.
but I'm not entirely sure.
So it's just basically having like adoptive cell therapy.
Yeah.
Okay, activated T cells. Cool. Well, thank you so much.
Any more questions?
All right. I think that about does it. So everybody go ahead and say thank you to Alyssa, and you can clap. Thanks, Alyssa. Clap into your microphone.
Thank you so much, Alisia. It's nice presentation.
Good. Yes, that was very impressive.
Guys.
All right. We have another deep dive on Wednesday. Or is it research? Pretty sure it's deep dive. Who's going on Wednesday?
Me?
Nima. All right.
I am present.
That's right. MRI contrast agents. Okay. Great. All right. I will see most of you then on Wednesday. Bye.